This protocol to purify the enzyme tryptophan synthase from Salmonella typhimurium is fast, simple, and short. Ammonium sulfate fractionation and size exclusion chromatography make it possible to purify the wild type and mutant tryptophan synthase complex in a single day. This technique can be applied to purify other related proteins, since ammonium sulfate fractionation and size exclusion chromatography are commonly used to purify non-tagged proteins.
To begin, disrupt the cell pellet by sonication on an ice water bath at 80% amplitude for 20 cycles using 10-second pulses and 20-second rests. Centrifuge the cell lysate at 30,000 times G for 30 minutes at 25 degrees Celsius. And aspirate the supernatant without dislodging the pellet. Filter the clarified supernatant fraction with a 0.45 micrometer filter at room temperature. Slowly add small amounts of ammonium sulfate to the filtrate until 20% saturation is reached. Carry out ammonium sulfate fractionation at 25 degrees Celsius, and gently stir the solution for 10 minutes, taking care to avoid bubbles. Centrifuge the solution again for 10 minutes, and transfer the supernatant into a clean flask for further use. Gently resuspend the pellet in 20 milliliters of sample buffer, and prepare a sample to run SDS page.